with everything going green in St. Louis, looking at green architecture, heard about and seen the LEED building certification going on. So Bruce Lindsay, who's the Dean of the College of Architecture at Washington University, is going to talk about some of the work that the other series that the Academy of Science is involved in, and just this year, is our conservation conversations, which tonight would have been the last one, except that we were approached by Mizzou, the Tigers, and we have a very special edition that is going to take place on Tuesday, April 13th. Laurel Neame is an environmental journalist, and she just published a book called Animal Investigators, Wildlife CSI. There's one lab in the U.S. or operated by the Fish and Wildlife Service that looks at the trafficking in illegal wildlife. And this is a different take on your television show, CSI. So out front we have some of these flyers that if you want to pick one up at the welcome desk, it'll remind you of the program on April 13th. So for tonight, Bob Merz is our speaker. He has been at the zoo since 1982. Let's do the math there, Melissa. But Bob has been full-time and started in the primate unit in 1980s, and he's been with the zoo for 20 years full-time, and has been at the Invertebrate Unit, actually even before we built the new building, for the last 10 years. And Bob has been the Zoological Manager for invertebrates and has taken a major role in the Conservation Center looking at the burying beetle. And raise your hand if you're a fan of Tom Waits. Okay, we have a few people here. So Bob is, brought in one of the more poetic, subversive musicians of our time to talk about probably one of the more unusual invertebrates, the American Burying Beetle, which is endangered, and we've been involved for a number of years doing both surveys and helping with conservation work, breeding and raising them in captivity and then releasing them in Ohio. So I'm going to turn it over to Bob, who will give us an update on the undertakers of the invertebrate work.
Thank you for showing up tonight. It's cold out and the games are on, and it's Mardi Gras. So I brought some beads. I'm going to throw them out if I see you falling asleep. So there we go. So if I see you dozing off, I'll toss them out. Here you go, Jim. You guys get some too. There you go. Okay. But if you do fall asleep, I will toss them out to wake you up. I'm going to speak from the microphone, and I'm going to look over this way. Can everyone hear me okay? Okay. I'm going to start. Let's go. I was debating on how to start on this. Invertebrates, it's a large topic, and where do you start? Do you start with beetles? Do you start with invertebrates? Do you go back to, and then the earth cooled? I don't know. I don't know where to start. So I decided to start, let's see here. I decided to start with taxonomy, because this is exciting stuff if you're an entomologist. But just to touch bases real quickly, invertebrates are animals, right? Plant, animal, mineral. Vertebrates are animals. Insects are in a group called arthropods. Arthropods have the jointed legs, so they're not worms, they're not leeches, but they're related to spiders and so on, crabs and whatnot. So from insects, we have different orders. You have different groups of insects. You have butterflies are some that people know. Beetles are other ones. Diptera, those are flies. You have those. But you know what? All that's kind of boring. And only real entomologists, and only a certain group of real entomologists find that fun. And that's true. That is true. But this is the fun stuff. Invertebrates rule. They do. If you take all the animals on the planet, everything, all the animals together, 98% of those are going to be invertebrates. That means they don't have a spine. The ones that have a spine, which would be fishes, birds, all the mammals out there, everything, that's only 2% of the entire animal kingdom. These are amazing numbers. One thing, if you were to take our zoo and have it represent the animal kingdom, we would have where the Red Rocks area is. Does everyone know where that is? We would have the, I don't know, dragonfly harbor, let's say. If we were to, we would take the primate house and have that devoted to clams or something. All the other mammals, everything else, all the elephants, all the hoofstock, all the primates, all the birds, all the fishes, everything would be represented in a building the size of our insectarium. So you have to represent everything in there. It shows you, first of all, the task that we have, and I think we do a pretty good job of it at our building if you haven't been through there, but it shows you the task that we have to represent all the invertebrates, 98% of the entire animal kingdom in one little building. It's a difficult task, but those are amazing numbers. Here's one. This is kind of fun too. If you were to weigh all the animals on the planet, every animal, just take it and weigh it. Every elephant, every whale, every bird, every fish, everything out there, weigh them all. 10% of all that weight would be ants. 10%, the entire weight of all the animals would be ants. Another 10% would be termites. That's two groups from one order, the Hymenopteron, that represent 20% of all the animal weight. That's amazing biomass. These are huge numbers we're talking about, and it's hard to wrap your head around a lot of times, and that's a lot of bugs. So some more fun stuff. This is interesting. It's estimated that in the average urban backyard within a year's time, there's 10,000 invertebrate species. 10,000 invertebrate species. Again, huge numbers. Now, a lot of these are ones that we don't see. If you pick up a handful of dirt, you're going to find a lot of little things in there that are hopping around. You can barely notice with your naked eye. But there's a lot of invertebrates out there in the average urban backyard. This is a great one. It's estimated that spiders can eat up to 21 tons of insects per acre in 1 year's time in rural land. So think about that. Think about how much a fly weighs. And then try to add that all up in 1 acre of land. They're eating moths, they're eating butterflies, they're eating a lot of things. Now, wrap your head around this one. Think about the breeding potential of all those tons of insects out there. And you think about the biocontrol that these spiders are making in 1 acre of land. So the next time you see one, don't spray it with a can of Raid. Go up and thank it for controlling those insect populations, because we need those spiders out there. One of the 32 to 36 orders, depending on who you listen to out there, of insects. So the 350,000 to 500,000 species of beetles on the planet. It's amazing. It's hard to wrap your head around. So I like quotes, and you can see that from the first thing. I think that they say things in a way that I would like to say them, but usually don't. And this is a great quote, and it's attributed to JBS Halliday, and it's, if one can conclude as to the nature of the Creator from a study of creation, it would appear that God has an inordinate fondness for stars and beetles. And if you think about it, it's probably true. So beetles. Coleoptera, that's the order that they're in. Genera, that word where it says 8 genera, genera is the plural of genus, for those that don't know. I'm going to poke myself in the eye with a laser pointer here. So genera is, it's genus, but a bunch of them. So what we're talking about is the American Burying Beetle. That falls under this grouping here. So the genus Nicrophorus. So the genus Nicrophorus has about 85 species worldwide and about 15 species in North America. And this will become important in a little bit, and I'll show you. But you can kind of see how all these species, these are all ones we can somewhat find around here. They all look very similar, and they are. They're in the same genus. But that's important when we're out in the field to be able to try to tell them apart. So Nicrophorus americanus, that's the largest carrion beetle in North America, and it was federally listed in 1989. At that time, they only knew of one population, which was off the coast of Rhode Island, and an island called Block Island, and that was all they could find. Since then, we found more. And this is a very telling map. All the gray here is the former range of the beetle, and all these little white spots are where they've been found, where we have actual confirmed, records of the beetle being found throughout history. The black spots now, not counting this, just these black areas are where they're found now. That's it. They used to be in this whole range around here, and now these are the only places we can find them. 7 states. That's it now. So here's another little more detail. You can see the individual, counties where they're found, and I'll talk about all of this in a bit. This red county right here is the areas in Ohio where we have been reintroducing the beetle, and the yellow are the captive colonies that are held in zoos and one university right there. There's another great quote by William Shakespeare: "Good things of the day begin to droop and drowse, while night's black agents to their praise do rouse." Shakespeare had added 2 words to that. He wouldn't have talking about the burying beetle. He should have said, "While night's black and red agents to their praise do rouse." Here's the life cycle. This is what happens with the beetles. They go out in the field. They're out there. Something dies. It could be anything. It could be a bird. It could be fish. It could be a snake. It could be a small mouse or something. And it needs to be of the right size for these beetles, between 100 grams and 200 roughly. What happens is a pair will find this beetle. Sometimes more do, and then they fight over it, and the one male wins and the one female wins. Sometimes other beetles come in. But what you end up with is one pair, a male and a female. They find this animal, and they can move it. They can move it up quite some distance, and they move it by laying on their back. And I will show you this in a bit, but they lay on their back and they use their feet, and they push it over them. They can move it some distance. They dig a hole underneath it. The prey item falls into the hole. That hole could be a foot or so deep. And then they cover that back up. While they're doing this whole process, while they're burying and moving, they remove fur if it's a mammal, scales if it's a fish or something, feathers if it's a bird, the beak, feet, so on. They remove all that. They take out bones, and then they form it into this meatball. And then they let that drop in ground, and under the ground they spray preservative, oral and anal antibacterial secretions on it, and that keeps it from decaying, and then they cover that up. Now what that does is that effectively removes it from the rest of the environment, so other things don't eat it and take advantage of it. They bury that underground. You can see they're underground in their chamber here with their little babies, which are these white worm-looking things. um, actually grub worms is what we call them. And this grub worm is about 10 days old or so. So they bury that. The grubs will sit up and beg for food, and I will show you that. The parents care for their young. They bite off pieces of this food, they go over, and they regurgitate it back into the young's mouth. Much like baby birds will sit up and beg for food, much the same way. So, and then in about a month or so later, all the new beetles come up. They'll pupate. So much like a butterfly is a caterpillar first, these beetles are the white grubs first, and then they basically change into that. They pupate over. Okay, let's see if this works. This is the film footage from beginning to end when they find it looks like a shrew or a vole of some sort. So here we go. They're moving the animal, and you can see they're going to move it, you'll see down here, by laying on their backs and using their legs. You can see them doing that. It's amazing. And they can move it quite some distance. They do this all in one night. Here, oh, it's beetle love. Look at that. Beautiful thing to behold. They will mate. They remove the fur. They dig the chamber, and then they bury it in there. And here they're preparing the carcass. And you'll see the meatball. This right here is the meatball. Then you'll see the young sit up and beg for food. Here's some of the young. And there they are. They're feeding their babies. Isn't that darling? They are a writhing mess. I love them. Phoretic mites. Phoretic mites just mean that these mites basically hitch a ride on the beetle to the next dead animal. These mites eat small egg. They'll eat fly eggs and also small maggots. Not a whole lot is known about them, but they're all over the beetle. I'm going to show you a picture, which is kind of gross, but you can get the size of the mites. They're tiny little guys because that's the back of one of these beetles. Here we go. That's an interesting picture, isn't it? And it gives me the heebie-jeebies. I'm scratching myself right now. What will happen is that the mites are all around, and then the male usually leaves the brood first. As the young start to get older, the young will go off on their own way, and then they turn into the beetle. What happens is right before the male leaves, the mites somehow know there's some sort of cue, and they all hop on the male. The female has a few on her, but not a whole lot. And then in a few days, the female leaves, and all the mites hop on her again. We're not really sure what the cue is, but we might look into that. So they hop on the female, and a few days later, and they can get covered with these mites. Now the next picture is interesting. The Roger Williams Zoo sent photos of some of the mites that they took under a scope to get them positively identified. They sent them to some guy that knows his mites. And what did they find? But they found mites on the mite. So we're talking these tiny little mites, and here I'm going to zoom in a little bit, and you have other mites living on that. We haven't really zoomed in on these mites yet, so who knows how far this will go. We'll see the future. So how do you determine if it's a male or a female? Because you have to start with a boy and a girl, right? So in the lab, this is what we do. We have to determine what's a male and a female. And it's a very easy process. And I'm sure after seeing this, you can all follow along. All you do is apply pressure to the abdomen, the dorsal side of the abdomen, just under the elytra. So here's where it gets really easy. Intracellular, I'm joking with you. We actually have a shortcut with the ABBs, as we call them, the American Burying Beetles. So here's the ABB shortcut. Pretty easy. What we're looking at is the face. I might shoot myself in the eye again. We're looking at the face. This pretty much is the same, this marking is the same on both male and female. Males, though, have that rectangle, and females have the triangle, and it's almost always right. There's some there that's a rectangle, kind of triangle looking thing, and we're not quite sure, but for the most part, it's always a rectangle triangle. Okay, so let's look again. We got male, right? And female. Okay, that's on a real beetle. Now, what are we looking at? Who can tell me? Female. Hey, someone gets bees. Hey, there we go. Yes, female. That's right. So that's what we do. We pair them up that way, male and female. It's pretty easy to tell them apart because the bearing beetles gave us that nice little shortcut. So what we do is we start with a quail, is what we use to raise them in the lab. And remember, it's going to be about 100 to 180 grams is what we use, somewhere right around there. We put them in a bucket with hard-packed soil, and we put the dead quail in there. They will bury this overnight. We come in, sometimes they're all the way down, to the bottom of that 7-gallon bucket, I guess is what it is. The feathers and the beak are up on top. Sometimes the feet are. There's some bones up there. The eggs hatch in about 3 days, actually. And then the parents will feed these babies underground for about, 10 days or so. That's what we look. So then we actually go in after that time. We excavate it up. We dig that up, and we make sure that we got the babies in there. And when we do see babies, we kind of count them, and then we check and make sure everything's fine. And then we'll allow them to move off on their own. Sometimes we'll help them by poking little holes into the soil to allow them to migrate away. And even if we don't, the female usually hangs around a bit longer than the male, and she'll do that. She'll kind of start holes for the babies as well. They pupate for about a month or so, and they form this earthen cell around themselves. And they do all of this underground. And then the new beetles emerge from the soil. And, okay, here's another quote break. When we try to pick out anything by itself, we find it hitched to everything in the universe. And that is so true. This actually leads in well to the next section here. So what's going on with the beetles? You saw the range map. They used to be all over the U.S. And now we're not finding them hardly anywhere. And we've been looking hard. You're going to see that in a bit too. One theory is that fragmented habitat is out there. So now we have more hedgerows, we have more strip malls, we have more roads, we have more housing developments. We have things that divide up their former habitat. And what you end up with, and you can see it all in here, this patchwork that you see when you fly in planes. You see that all the time. But what you end up with is this edge habitat. And the edge habitat is very favorable habitat for other scavengers. They do very well in there. So you have a burying beetle that finds a dead animal, it buries it, lays its eggs, it might even raise its grubs for a bit. One of these guys comes along, and they can smell very well, and they dig it up. They dig up the old animal, they dig up the beetle grubs, which are added bonus, actually, and then they eat them. So that might be happening, but we're not sure. Really not sure. Remember I said that the food size has to be the right size, has to be between 100 and 200 grams. Well, these animals, who knows what they are? Anyone? It's the prairie chickens, right there, prairie chickens. They used to be found all over the range, and now they're very hard to find them anymore. In fact, we're reintroducing them into our state from a group, I think from Nebraska, but I'm not positive on that. And then this is the passenger pigeon, which would have been perfect size, and that died off in the early 1900s. In fact, I think you can see the last one that they had in captivity at the Cincinnati Zoo. They have her taxidermied. And for 200 years we've been conquering nature, and now we're beating it to death, says Tom McMillan. Insecticides, that's been brought up as one. I mean, it somewhat makes sense, doesn't it? Insecticides mean dead bugs. And they do. The one problem with this theory, though, is that there's a lot of other burying beetles out there, and we are finding them by the bucketful. So if insecticides were hitting one beetle, why is it going after only one species and not the other ones? It might factor in, but it's not looking like that's the reason. But that's an easy one to blame. Like I said, we're finding other beetles. Here's this whole book on, carrion beetles in just one state. They are out there. We do find them. We find hundreds of them, actually thousands of them, and I'll show you that in a moment too. But they are out there, so it could be they're just being outcompeted, that the conditions are more favorable for the other beetles and not as favorable for the ABBs. Walt Kelly, I think Jim often quotes Walt Kelly from time to time. We've met the enemy and he is us. So the other reason could be light pollution. This is a very interesting theory. Okay, remember that range map I showed you before? That's the U.S., that's the former range is the green, and then this is where they're found now in the brown lighting here, but in the brown counties. Now I want to show you something interesting. Here's a satellite image of the U.S. at night. Look at where all the light is mainly. And I'm going to put the range over it as best I could draw it. And where are we finding beetles now? Well, we're finding them in Arkansas, just outside that light, in Texas, in Oklahoma, in Kansas, in South Dakota, in Nebraska, and then South Dakota, and then just off the coast on Block Island. So all outside of that big range of light in there. It could be early studies have shown in labs that the ABBs get confused by two types of light. That would be the metal halide and the mercury vapor light. They don't run away from it. They just get confused and they don't bury the dead animal. The other animals do. So that might be one of the reasons. We're just not sure. Henry David Thoreau said, "In wildness is the preservation of the world." This is great. I love this. Zach Hoffman could work for the U.S. Fish and Wildlife Service because this is a plan that we should have for the beetle here. And he says here, help burying beetles. That's good. We're doing that. Don't keep them as pets. Well, we're not doing that here at the zoo. We are raising them, so we're right in line with Zach. I love this one. It's don't hit them with sticks. As far as I know, we haven't done that much. And then the last one is, if you're a zookeeper, breed them. So we're following Zach's plan, and that's a good conservation strategy, I would say. I love that. But what are we doing, honestly? What are we doing here at our zoo? We are captive rearing. Let's start with that. Since November 2004, with some help from the Beetles, we have produced exactly 4,902 individual beetles. Beetles are housed in their own individual little habitats, and then these are the buckets where they're being raised in. And we do note every single beetle, every beetle. So how have we done this? How have we managed over 6 years to care for 4,900 beetles? How have we done it? Is it the spectacular crew of zookeepers we have? Yeah, I guess partly that's some of it. Is it my staggering ingenuity, sparkling personality, and rugged good looks? I get that all the time, and humbly I would have to say yes, that probably is part of it too. But we have another trick up our sleeves, and that trick is volunteers. I often, who knows the story of Tom Sawyer painting the fence? I often feel like that with our volunteers. We give them this job where they're cleaning out these stinky, smelly beetles, feeding them like wriggly worms, and they're loving it. And don't know, it's just interesting. But we have a host of volunteers. I'm going to show two, but we have many, many, many volunteers that come in all the time. And they come in and they call them my beetles. These are my beetles. How are my beetles doing? And we could not do it without them. We honestly couldn't. So I want to give a big shout out to our volunteers. Thank you. Thank you. We even had a volunteer write an access database that tracks every single beetle. So we enter in the info when the beetle pops up. We know exactly who the parents were, who the grandparents were. We can trace them all the way back. We know how old they were. We know what their age they were. We can put their weights in there. We measure parts of their body and we put all that in there. And we keep all that as a record. It even prints the pinning labels after death. One interesting thing about this beetle is, since it's a U.S. Endangered beetle, we have to keep all the remains. We cannot get rid of them at all. So we have to maintain them, and we have to know exactly what beetle this was, or what leg this leg went, what beetle this leg went to, if the leg should fall off even. So we really need to maintain that. There was also an SSP, which is a species survival plan. That is all the zoos in AZA get together and say, we want to manage this beetle population as one giant population between all the zoos. So in order to do that, we have to know who's related to whom, and keep records on these beetles. So that was formed a few years ago, and I maintain the Studbook for ABB. So I keep a record of all the beetles in captivity, and from that we use that information to plan the breeding program for that. So we do not have to take them, from the wild. So that's what we're doing. We're also surveying the state of Missouri. That's just a rough map for a few years of some of the sites we surveyed. Here's how you do it. You place a bucket in a hole. You have to dig the hole first, obviously. It's pretty easy. Then you place a jar in there with really, really old, smelly, fizzy chicken. It's one of the most disgusting smells you've ever smelled. In that jar, you place a screen lid on top. We'll actually leave the bait, we call it bait, but we'll leave that sit out for 4 days or so in the hot sun during the St. Louis summer, inside a jar. We have to watch the jars because the gases will build up and they can blow up on you. So you have to vent the gases from time to time. It's a horrible smell, and it is. It's a retching smell. We place a screen over that. What we'll do is we have this chicken wire, and we have to kind of stake that down because a lot of things really want that old nasty chicken. I cannot believe that, but they do. And we have come back the next day often enough and have found the traps all torn apart and the bait jar ripped to shreds and drug off across the prairie. So then we place a shingle over the trap. That's usually a piece of wood, and that protects it from the sun and rain. So, because we don't want the beetles to bake if they get in there. Here's another quote break. I say, if your knees aren't green by the end of the day, you ought to seriously re-examine your life. Well, when we set these traps, because we do this over and over and over again, and it's not just one trap, we'll set 10 at a time, 20 at a time, and more. Your legs your knees are green and brown and smelly. So we take an exact GPS with it. That's important because if we should find something, we want to know right where we found it. The other thing that we do is we find a lot of other beetles in these traps, a lot of other things in these traps actually, and we write down everything that we find. And then we can report back to the people that manage the land what we found and where. And that's good for them to know what they have on their land. So we check the traps for several days and we record everything that comes to it. And you sometimes walk up and see a bucket like this. And actually I see 2 species right off the top of that in there. But we count and record every one of those. And that can be monotonous. So variety is not the spice of life. It's a pain in the neck when you're talking about burying beetles. Remember, I showed this earlier. All these look slightly different, and these aren't the best. Like some of those have turned dark on you, but they look very well. They basically look the same when you look at them in a bucket. And you have to pull them up. So how do we do it? There's about 15 types in North America, and we have to count and ID each of them. This is an easy one, but that one's much larger, although size isn't always the best way. This one doesn't have the orange on this little shield-like thing on its back. So we have little guides that we've put together that we pull those out sometimes, and we'll look at all the different bizarre little parts on the beetle and say, well, is it this one or is it that one? And we ID them that way. So here's what we've done. We've surveyed for 8 complete seasons. That's usually from May or so till about September, October. And we have not found any Microphorus americanus in our state. They haven't been found here since '82, but they have been found here within my lifetime. So that's interesting to note, that they used to be out there and now they're not. We found several other Microphorus species. Here's some data just to give you an idea of what we've done in the past year. So this past summer, staff from the zoo looked at over 40,000 individual sylphidae beetles, just one type of beetle, over 40,000, as well as 7,000 other insects. 20,000 belong to the same genus as the ABB. And then 4 species of burying beetle were found in our pitfall traps. 91% were a species called Microphorus marginatus, which is real common out there, around here at least. And then there were other beetles in the same genus, and these are some other ones that we find. But we do mark and record every beetle. This here is important for me to show. This is Rob Jean and his wife. They've been surveying for us for, I'm trying to think now, I think like four years now, 4 seasons, and they just go out and survey for us. And it's been great to have them do that. Rob is actually a bee expert, and he found a second love with the beetles now, which is nice to see that. And hopefully he'll be back this year. He plans on coming back. But you don't see him around here much because he's always out in the field. So people have brought up, are we going to reintroduce them back to our state? They were found here. They were found here, you know, within my lifetime, actually. And why not just put some back? Well, we might. We're actually working with the Fish and Wildlife Service to develop a plan to put them back. What you don't want to do is just start tossing beetles back into the wild. You really want to have a plan set. You want to make sure you do it right. You want to make sure that you're not putting things out there in a habitat where they might conflict with, land use or something, or private landowners, for instance. So you don't just want to tell, oh, The Simpsons, one of my favorites. Here's another quote, great. Oh, so Mother Nature needs a favor. Well, maybe she should have thought of that when she was besetting us with droughts and floods and poison monkeys. Nature started the fight for survival, and now she wants to quit because she's losing. Well, I say hard cheese. That's from Montgomery Burns, if anyone knows Mr. Burns from The Simpsons. And I'm so glad we don't feel that way here at the zoo, because we've been working very, very hard to bring this beetle back. So we have contributed for the past 5 years to a reintroduction project in Ohio, southeastern Ohio. So both beetles and personnel. So beetles that we raise here at the zoo, we bring to Ohio and we plant them back in the wild. And what you see here is a bee tag that we glued to the back of this beetle. We have stopped doing that, and what we do now, this hard shell on the back of the beetle is actually a hardened forewing cover. And so we'll actually notch that. We take a little thing that burns a little notch in it. And it doesn't hurt the beetle, and it will permanently mark the beetle. The bee tags, if you've ever tried, who's tried to glue a bee tag to a live beetle? Here, anyone? Okay, yeah, there's a few people. It's very difficult. It's very difficult. The bee tags don't always stick. These beetles, you know, they dig in soil, and the bee tags can just kind of scrape right off. So the notches are a real good way to positively ID the beetle. So we've moved to that, which is actually good for all of us. So far, we've contributed over 1,500 beetles to this project, to this reintroduction project. And these are some photos taken from the most recent one where we had some kids help us. And I was a little nervous about that. It turned out to be wonderful. It was just great. The kids really got it. They knew what they were doing was very important, and they knew to be very cautious around the beetles. So in 2009, we contributed close to 600 beetles, almost 300 pairs, and they were introduced to Roy National Forest somewhere in southeast Ohio. And here's one of our keepers, Tim O'Sullivan, explaining to people how to do this. Because you have a large group. And here's how we do it. We take the soil plug out of the ground. You take the soil plug out, and then you dig a little side chamber off to the side with a hand trowel. You place a dead quail in there, and you add 2 beetles, a male and a female. We have them paired up before we even go up there. And we drive up with all these beetles, and then you place the soil plug back on top. And then on top of that, you'll see we take the screen mesh, this kennel fencing, and we stake that down, because remember, a lot of things really want that old, chicken. So we stake that over the top just to give the beetles a chance. They come back in about 10 days just to check some of the broods, and from that we can tell how well it's done. And as you can tell, there's a lot of sweat involved and a lot of people working on this beetle. And I was taking the picture, so I didn't dig at all. I'll be doing it. PHVA is a acronym for a population habitat and viability study. This was actually a meeting that we held in 2005, and I won't spend a whole lot of time on it, but it was a very important meeting. We hosted it here, and we brought all these people from all over the U.S. And one from the UK to talk about how we're going to recover this beetle in North America. We also invited U.S. Fish and Wildlife, military, timber companies and so on, where that own land that have this beetle on it. And we talked to them about the best approaches to take on their land as well. So it actually worked out very well, and I think we'll move ahead the recovery of this beetle in the end. So this quote goes right into the next thing that we're doing. Human history has become more a race between education and catastrophe. H.G. Wells said that. Public education. So education, the zoo, is a highly respected zoo here. It was voted #1 by Vegas Parody magazine. So a lot of people come through the zoo, and there's a lot of potential for them to be exposed to this project and to the beetle. We're also contributing to the species survival plans education component as well. The zoo is free. It's open to everyone, and about 3 million people come to the zoo every year, and about 1/3 of that goes through our building there and has the potential to see the project and learn about what we're doing. There's also a website that's focused on the recovery effort that's hosted by our zoo. So the website is for people that are working directly with the project, and they can find out what's going on, in different areas, so on. We also had the zoo show, and the reason I wanted to show this picture is I had hair at one point. I just wanted people to notice that, if you don't remember. I did have hair. But the zoo show does reach quite a bit of people, and the beetle has been featured on the show as well. So unless someone like you cares a whole awful lot, nothing's going to get better. It's not. And that was from the Lorax, Dr. Seuss. And that's where PR factors in. It's a difficult thing to make people care about bugs. At times it can be, but this beetle actually has a bit of a personality to it, I think, because it's large and black and red and big, and it does weird things with dead bodies. I think people somewhat like it. So this project was voted the #2 conservation success story in 2006 by AZA. What that has done, though, within the zoo community has raised a lot of interest in the project, and a lot more zoos are wanting to get involved and help in all different ways. Some zoos might not be able to have the animal, for instance, but they can contribute to the education component, for instance. So that's happening, which is nice. Also, Jane Goodall, if anyone has seen her new book, she has a whole chapter in there devoted to the project, and she mentions our zoo in it. And by project, I'm talking about the nationwide effort, to recover the beetle. But she does give us a nice plug in there, which is good. Theory guides, experiment decides. How many times have we all said that? What we are doing is participating in a bit of colony research. So there's a brood ball survey where we're looking at these little worms that kind of live on there, because we don't know if they're good or bad, if they help, if they hurt, if they don't matter at all. We don't know. So we've been helping with that, and that's being conducted by the vet department at the Roger Williams Zoo. This is real baseline data. We really don't know a lot about these beetles, and the more baseline data we can get, the better off we'll be at the end. Genes, how related these beetles are to one another is something that we don't know in the wild. So what we're trying to find out is what's the current effective genetic population size. That basically means is how related are all the wild beetles to one another, and is it basically going to work out there or not? And was there or is there a bottleneck going on, or did one happen at any time in history? And then if there are different populations, is there any interbreeding? Is there any contribution from one population to the next happening in the wild, and if there is, how? So what we're doing is, through the University of Austin, we are getting secretions from the beetles on this paper, and then that will be analyzed, and we'll compare the genetics of all the wild populations out there, all the ones that we have in all the zoos and so on, and compare them to one another. The interesting thing is legs from specimens from universities and also natural history museums and so on will be sent to this lady. She can extract the DNA from the leg without hurting the leg, put it back in the mail, send it back to them, and they can glue that leg back on their pinned animal. What's nice about that is the pinned animals all have a little tag on there that says when it was found and also where, so we'll know what county it was found in, and also when, and we'll be able to track if there was any sort of genetic bottleneck throughout history, and then that will actually be interesting, because then we'll be able to go to that spot and find out what was happening at that time in history, and maybe get some insight into that. I mentioned that we were clipping that hardened wing cover on the beetle. Well, we do know that the beetles, when they feed their young, will call their young over using their wings. They move their soft wings basically underneath there, and it makes this little, it's a little sound that they make. So we wanted to make sure that that's not hurting the populations of beetles that we're putting back into the wild. So there's a acoustic study being done where we recorded all of that before they were clipped and then after they were clipped, and then we're pairing them up to determine if we have more offspring from ones that haven't been clipped or so on. What's interesting, and it's hard to see here, is that right about, this is working, right about there is the range of human hearing. Everything above it is out of our range of hearing. So there's a lot of sound up here that is definitely being impacted when we do that. And, whether that has some impact or not, we're still gathering that data. The male beetles, when they find a dead animal, will do this headstand and send off this pheromone scent that calls all the other beetles in. We really don't know what makes that up. So we are collecting that pheromone on filter paper, sending that off to be analyzed. And the potential of that is you could potentially make a bait that will bring the beetles to that bait. So that would be important in a situation, for instance, where they're going to build a bridge or something, and they need to bait away the beetles to a different habitat, and then they can go through and build the bridge. So that would be one of the situations. Also little's known about their habitats in the wild. We find them at traps that we set, so we know they can go this far, each night, let's say, but all we know is that they're going to that trap. We don't know what they would do if that trap were not there. So we might find them, 2 miles away, but maybe they could fly 5 or 10. We just don't know. We're finding them, at certain traps. So what we can try to do is to mount a little backpack on there and monitor them in the wild. Now this is a little big and looks a little silly on the beetle, and in fact it is, but what we have been able to do is to test this in the lab, and then from that testing determine that it would not work in the wild and force the company that makes them to try to make it even smaller. So hopefully we're going to get a smaller one soon on that. So what we have here, what we're doing is a complete program. To some extent, we had the captive breeding, field surveys, the field research, captive research and reintroductions, conservation, education, and PR, and it all fits on one sheet. And those are our partners. We're getting to the end of the talk. Anyone sleeping? Do I need to throw beads? Questions? He asked, what's the lifespan of the beetles? Is that what you were asking? They can, it depends on who you talk to. We've had beetles live for almost a year as a full-grown adult. Typically in the wild, you would say a few months, like maybe 3 months or so, and we like to say 1 season. So about that. You always have some that live a bit longer, but I would say about 2 or 3 months is a good lifespan. Yeah. Mm-hmm. Yes. Nita asked about the spraying with the antibacterial. They actually they kind of leak it out more so than to kind of spray it, but they leak it out and they cover the dead animal with this, and it keeps it from getting all gross. And Okay. They like it a little rotting, but it needs to keep for about 10 days, so they have something to feed their babies. Yeah, right up front. No, yeah, they split up. The male leaves about 2 or 3 days before the female, and then the female leaves later. I mean, it is possible they could find, find the same, male or female, again, some night, but they basically just pair up to raise the babies, and that's it. Yeah, there was one way in the back. Yes, sir. Yeah, there was one at the University of, I want to say Nebraska, and it was just a real simple one where they had a bucket with a dead animal in it, and then they had a male female over here and a male female over here. They let them come into the bucket, and they exposed the dead animal to certain lights. What they found was that the ABBs would not bury the animal. They just didn't know what to do. They just kind of walked around, and they kind of wandered off. The other burying beetles, which are actually more active in the evenings, were fine with it. That didn't seem to bother them. So that was one small little thing that was being done, and I haven't heard anything since then. It was, yes, correct. No, there was a just recently, Phillips Light Company put out something about that, and that's being passed around now. So there may be funding to really look into that, and I hope they do. Phillips is concerned about the impact of lights on threatened animals, is the way they put it, and so they want to fund projects that would, kind of look into that for them. So someone may do that soon. Yeah. Pardon? Corporation, yeah, they had put that out there where they would, yeah. Phillips had. That happens from time to time. That does happen. Yes, sir. Mm-hmm. So with the icebox effect, could you explain that? Yeah. Oh, okay. Right. 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 Yes. Yeah, and so is that a goal? Is that what you're asking? I'm sorry. It has. I mean, habitat is always. I think the one of the main thrusts now in conservation is more the ecoregion approach, where what you're looking at is what's healthy for these habitats within this region. And so, yes, it is being looked at in that level because we're not really focusing necessarily on the beetle. We're focusing on that whole ecology within it that the beetle is just a part of. So, yes, it is, but it's a difficult thing to do because there's so many factors, and we honestly don't know even where the beetle goes. You know, when it gets cold out, we're not really sure. We have ideas, but no one's really tracked broods out there in the wild. So what we're trying to do is to first of all get the baseline info, and then try to approach it from that eco-region approach. And maybe, you know, yeah, I think the other side of that is they have supplemented some reintroduced populations with dead animals. They toss them out there for the animals, whether that helps the beetles or whether that helps the things that also eat as well, as you know, sort of up in the air right now. So it gets kind of confusing when you talk about whole areas, because we kind of don't know what's wrong with the whole system. Does that answer your question? Okay. No, not really. We do know that they do eat fly eggs, which certainly will help. That will reduce competition for the ABBs. The ABBs seem to be able to reproduce without mites. I mean, we have had times where we've taken mites off, or the beetles don't have mites, and they can reproduce. Whether that would go on for generation after generation or not, it's hard to say. My gut feeling is that they're helpful, to the beetle. That you need them, they're always found with the beetle. Whether they're the same mites or not, we don't know. There could be different mites from different regions, so that might be the case. But we haven't really looked into them a whole lot either. Yeah. Anything else? Happy Fat Tuesday, and thank you for coming. I got beads.